So before we get started, um, just one quick thing to mention. If at all possible, um, if you have the opportunity to contact me during Learning Block 3, drop in, ask your question in the Google Meet. That would be ideal. That would be the best way to do things. Um, I'm getting a lot of emails after hours. Many of them are past 8 p.m. Just try and keep in mind that if you're sending me an email after 8 p.m., I'm probably not going to get to that same day. That's way outside of my communication for work hours. So I'll do my best. If it's after 8 p.m., you're probably not going to get a response. I will look at them the next day when I have an opportunity. But try and make sure you're communicating with me during that learning block 3 if possible, because I think what's happening is some people are saying, well, I have this question, but I'll just ask it at 5. That's nice. I gotta look after my kids. I'm not just like on email all day just answering questions. So I mean, I'll do my best to get back to you. It's not that I don't care. I do care, but just please be aware that it would be ideal if you could ask those questions a little bit earlier in the day. That would be helpful. And ideally during learning block 3, where I'm devoted all of my time to answering student questions. And so just something to mention. Okay. That being said, yesterday midnight was the due date for most of the stuff from Learning Block 1. I noticed some people mentioned to me that I forgot to change the quiz deadline to yesterday at midnight. I fixed that. So now the quiz deadline is now yesterday at midnight. So if you did, it won't show up as late or anything. I fixed all that. That was my mistake. I fixed all the other quizzes too. I had it set to the last day of the unit instead of the day after. If you are in a position now that you're not finished, one of the major assignments, which would be the vocabulary list, the species spotlight assignment, or the quiz, Trent, just email me with that stuff, please, okay? If you wouldn't mind sending me an email. I don't communicate over the chat with students about extensions and stuff. Just that's a private conversation that we can have, okay? So send me an email. Just make sure that you are communicating. Sure, that's great. Make sure that you're communicating with me if you're in that position. You send me an email if you're not done. And when you send that email, you should propose an alternate deadline for yourself. What do you believe is a fair deadline? It'll just speed the email communication up, because the first thing I'm going to ask you when you email me is, what do you think is a reasonable timeline for you to complete it? And I don't give you a reasonable timeline because I don't know what your situation is. Do you have a chess tournament coming up on that day or something? Like, I don't know, whatever your personal life is, I don't know. But you propose what you believe to be a realistic, fair alternative deadline. And if I agree with you that that is realistic and fair, then I'll tell you, sure, get it in by then. If I don't, I'll also tell you that, and we'll try and renegotiate something else. Because, and again, keep in mind that my motivation there is to keep you from falling behind. Keep the deadlines reasonable, but also not extend them so far that you're going to run into trouble with the next unit. So, because I want you to get the credit. So, and I've seen bad things happen already this year to students getting behind and stuff. And this is an extremely content-rich course. There aren't a huge number of assignments, but it's very content-rich. So it's not like there's nothing to do every day. Like, there's lots and lots of stuff we're doing. So anyway, my objective here is always for you to get the credit. I want you to be successful. So please contact me. Don't just leave it and let it go if you haven't handed something in within the deadlines. Okay, once I start having to track you down or whatever, we're going to get into a bad place. Don't make me track you down if you don't have something important handed in. Just let me know, okay, and we'll negotiate a deadline. That's the responsible thing for you to do as a student, and you better be learning how to do that. In university, some people will tell you that there's no extensions and things like that. That's not true. You can occasionally get extensions from profs. Now, there are profs that don't give you extensions, But you can get extensions for things if you have a valid reason to require an extension. But you have to ask for them. If you don't ask for an extension in university, uh, you just get zero. That assignment is done for you. It's over. Um, and you have to learn how to be responsible about your stuff if you can't get it done on time. That's going to happen in any job you're ever going to have. I mean, you can't always make a deadline. It will happen to you. Now you have to make the vast majority of your deadlines, because if you don't make your deadlines, you get fired. But in any job, and by the way, that's true for my job too. I have to get your report cards in on time. I have deadlines, like real deadlines that exist, and I have to meet them. And I can't just like send a little letter in and say like, oh, I didn't get the report cards and I didn't feel like doing them. Like it doesn't that doesn't exist. That's not a real thing. And so the same thing is true for like literally any job you're going to have. You got to meet deadlines. Um, and if you can't, you got to make a reasonable attempt to make them, and discuss with the person who you're handing the material into that you need to do that, or that you're going to do that. So just get in the habit. Get in the habit of being responsible for your deadlines. Um, okay, awesome. That's fine, Trent. This wasn't directed only at you. This is directed at everybody. I've received many emails about this. And the reason I mention that is because we used to have really hard deadlines in education, which I don't necessarily agree with either. We used to have deadlines. I mean, we could really make our own rules. When I started teaching, if you didn't want to accept assignments after the deadline, you didn't have to. You could just say, well, too bad, you missed it. Which I don't think that's a good policy personally, because everybody has their own personal life circumstances, and there's reasons that people don't have stuff done on time. And there are teachers that set deadlines that are unreasonable. That also exists. I try not to do that, but it's possible. So I don't think that's a great policy. But we did used to have consequences for people handing stuff in late. You guys probably aren't even aware of this. This changed before you went to high school, I'm pretty sure. But we used to take marks off every day something was late. Most schools did 10% a day. Some schools did 15% a day, up to a maximum of 50%. At which point, if you don't hand it in when it's worth 50%, then it's worth zero. So then it just becomes an assignment you can no longer submit. I don't know how I feel about that. It was nice that it provided students with motivation to hand things in, but the problem is, in general, it just made students not get the credit, because they would hand something of very poor quality in late, and so it's already worth 50% of whatever you earn, and so then it's usually worth 10% or something like that after you mark it. So that may not be the best system either, but at least there was some motivation for students to hand things in on time. What we're operating with now is a system of pretty please. Can you please hand these things in on time so that I can mark them within a reasonable amount of time, provide you with feedback so you can learn something in this course and you are likely to get the credit. Like, that should be motivation for you, but it just isn't for some students. So sorry, that's my frustration. I do want everyone in here to be successful, and I just feel like you're causing yourself a great deal of trouble by just not doing things that you need to be doing on time. Again, if you're getting things in a day late and you're letting me know about it or whatever, it's not a huge deal. It's not problematic. I'm not trying to give people a hard time, especially the people that emailed me and made accommodations. Good for you. Thank you for emailing me and doing that. I really appreciate that. It's the students that owe me things now that have not communicated with me. So that's what's more problematic. So if you're in that boat, please do it. Okay, communicate with me and be responsible for the things that you need to get done. I want you to get this credit. Okay, I'm gonna stop talking about that now. Sorry, that's my little diatribe on handing things in late. So yesterday we did some review, and we also did some new content. So we talked about, oh wait, sorry, no, the review was the previous day. You guys didn't do protein synthesis in grade 10. Turn my mic up. Oh, that's so funny. Yesterday I had to turn it down because it was insanely loud. Okay, yeah, I can turn it up, no problem. Mic check, mic check, mic check. Now I'm turning the gain way up. Mic check, mic check. How's that? I've turned the gain up quite a bit. Better? Okay, awesome. I don't want to turn it up too much because then I'll max out the mic and then it's very uncomfortable for people listening. I wonder if I changed the positioning of this or something? That's weird. Okay. Move it a little bit closer to my head too. All right. So yesterday we discussed the process of protein synthesis, also known as the central dogma of biology, the most important concept really in biology. This idea of how all living things produce their functional structures, proteins, from DNA, from their body plan. We did a review of mitosis, which hopefully looked somewhat familiar to you. I really would prefer if you knew it extremely well based on what we're going to do today, but I mean, people will be in different places regarding that. Hopefully it's quite familiar to you at this point. Interphase, prophase, metaphase, anaphase, telophase. You should be able to picture with each of those phases what happens in that phase. If I said metaphase, oh, that's when the chromatids line up across the center of the cell. Interphase, that's when the DNA is replicated, that kind of stuff. If you don't have a general sort of plan for that, well, this is going to be a little bit more difficult for you, what we're going to do today, but we'll do your best, do your best. What we're going to do is build on that idea of mitosis today and add a new process to your lexicon of biological information. And that process is meiosis. So meiosis is building sperm and eggs. Hopefully people had an opportunity to watch this summary video. The course notes that you're going to need right now are right here. They're actually on day 6 page, even though this is technically day 7. And so you're going to need that, and you're going to need to open up those course notes right there. So if you haven't done that already, those are the course notes that you need in CAMI. And we're going to discuss this process in a great deal more detail. Before I get started on that, so that's what we're going to do right now. Does anybody have any questions about the topics that we covered yesterday? Any burning questions? Protein synthesis questions or mitosis questions? No? If any pop up in the chat, just let me know. Okay, so there's the note right there. In the diversity unit, one of the things we talked about was this idea that diversity, and the one that we're going to be looking at specifically here is genetic diversity. So that is diversity in the actual genomes of a species. That is, if you were to look at all the different genomes for all the different people in the world, I'm using people as an example, there is variability or diversity in their genomes. They're not exactly the same. Obviously, you're not genetically identical to your parents or to your brothers or sisters or neighbors or anyone, unless you have a twin, an identical twin. That diversity is important for our species' survival because they're going to be members of the species that are more tolerant to heat. They're more tolerant to drought. They're more tolerant to wet conditions. They're more resistant to a particular fungus. They're more resistant to a particular bacteria. They're more resistant to a particular viral infection. You name it. So variability in any species is important for survival, and that's sort of like the main thrust of the diversity unit, this idea that diversity equals survival. In sexually reproducing species, which if you look at the animal kingdom is most animals reproduce sexually, and it's not just animals, by the way. Most plants reproduce sexually. Most fungi reproduce sexually. Most protists reproduce sexually. And bisexually, I mean that there is a male and a female, and that they each produce a sperm or egg, and then those two come together to form their offspring. That's sexual reproduction. That is by far the default in nature. It is the most common method of reproduction. And the reason for that is that sexual reproduction produces variability. And like I said, that's a theme that you're going to see throughout the course, that this is diversity equals life, diversity equals survival. Sexual reproduction creates new variations because, and this is should be fairly obvious for you to see, you are not the same genetically as either of your parents. You're actually a mishmash hybrid of the genetics of both of your parents. So if your parents were producing genetically identical offspring, that would not create additional diversity within the species, genetic diversity, which does not lead to greater survival. So as an evolutionary trait, evolution has been selecting for species that are creating diverse offspring that are more likely to survive. That is just a product of the evolutionary process, and which is why sexually reproducing species are by far the most common. Now, there is an advantage to reproducing asexually, which is that it's easy. It requires very little energy. You don't need to find a partner. You just need to make an exact copy of yourself. And so it's very low. It's not very energy intensive, that process. Whereas sexual reproduction is very energy intensive. You got to produce gametes. You got to find a partner. You have to mate. There's all kinds of partner dynamics that are involved in sexual reproduction. All that is skipped in asexual reproduction. So there is an advantage to asexual, which is why those species exist. However, when push comes to shove, sexual reproducing organisms almost always outcompete asexually reproducing ones if there is ever any adversity, any difficulty in their environment. Then the sexually reproducing species always take over at that point. So anyway, we have a distinct reproductive advantage because we are asexual, or sorry, not asexual, sexual reproducing species. I have a little list of terminology there. One of the words I crossed out was this idea of non-sister chromatids. This is like a made-up term. This doesn't really exist. I'm going to call them non-identical sisters. You'll know what I'm talking about in a second, but I can't remember if I actually cut that terminology piece out. But if I didn't, I would cross out non-sister chromatids because that's not a real term. That's like a made-up term. So this is a shared note that we usually, all the grade 11 bio teachers use to try and keep things consistent, how we teach the process of meiosis. But I don't like that word, and it's not real, so so I cross it out. Anyway, what you need to do in order to sexually reproduce, as I mentioned, is that you need to produce something called gametes. Gametes are also called sex cells. Those are your reproductive cells, sperm or egg. And the purpose of making those cells is that you have to create a cell with half of your genetic information. And the reason it has to be half is because your partner is going to contribute the other half, and then you're going to make an offspring with the full set. So you may remember that humans have a diploid number. Diploid is the full set of chromosomes of 46. That is the full number that you have. You have 46 chromosomes. So if you're going to make a gamete, a sex cell, you have to cut that number in half. So half of that number is 23. That's known as the haploid number for us, for humans, or the haploid number in general is just half of your total complement. And the reason that you have to give half is because if you gave more than half, then you would end up with an offspring that has more than 46, which is going to cause trouble. We're actually going to talk about what happens in particular circumstances where you get more than 46. Somebody mentioned Down syndrome the other day. That's one example of a problem that can occur when you have too many chromosomes. But you can only have 46. 46 is the full complement, and you don't want more. Okay, so we call that our diploid number. Our somatic cells, here's a new, I don't know if somatic is actually a new term for you. Probably is. I doubt you talked about it in grade 10. But somatic cells are adult cells. So your entire body, every cell that has a nucleus in it is a somatic cell. That's an adult cell. And all of those cells have the diploid number in them, except for red blood cells and a couple other edge cases. but almost all of them have a nucleus with 46 chromosomes. Question: if the gametes are half the DNA, do we sometimes resemble one parent more because their genes are more dominant? Yes, absolutely. That's very insightful, Kim. That is exactly what happens. We're going to talk about the idea of dominant and recessive a little bit later in this unit, but yes, one parent can contribute more dominant genes than the other parent. Even though they're only contributing half, 50% of the genes, one parent might have more dominant ones than the other one. And yes, oftentimes if those genes are related to appearance, which they are often, then that parent, your baby will look more like that parent. My brother's kid looks, in my opinion, exactly like him, although he did have a girl, but it's like a girl version of him. I think it's funny, but she looks really exactly like him. That's my opinion. I don't think she looks very much like his wife. Our son, I think looks like my wife. I don't think he looks like me, but anyway, obviously that changes as children age as well. So sometimes they later in life they look like the other parent or a weird hybrid of both. I look exactly like my father, so like you could put a picture of him right next to me, and I really look exactly like him. I look absolutely nothing like my mom, so which is interesting. So his genes are clearly more dominant in terms of appearance. Yes, Trenton. Down syndrome is you can think of it as a chromosome overdose. It's when you have an extra of one particular chromosome. We're going to talk about some of those cases later, so I'm not going to get into detail now. But yes, that is what Down syndrome is. Okay, so if you were to take a somatic cell, remember somatic or adult cells, from a male and female of a particular species and put them together, so two full sets, you would end up with 4N chromosomes at the end. 4N genetic information, a 2N and a 2N that are completely different from each other, and you get a 4N. 4N is not compatible with life. There is no way to produce a human with 4N chromosomes and have it survive, so it's not possible to do that. There are organisms that allow it. So it's weird. Biology is all about the exceptions. But there are plants that allow extreme polyploidy. Polyploidy is when you have poly, remember, means many, right? So polyploidy, instead of being diploid or haploid, polyploid means that you just have many chromosomes, many more than the set that you're supposed to have. It can be one additional, and you'd call it polyploidy, but it can also be more than one extra, and that's called polyploidy. And plants are very tolerant to polyploidy. There are some plants that will just allow a full bonus set of chromosomes, and the plant can still survive and reproduce normally. So there are some unusual cases in nature where there are organisms that are tolerant to polyploidy, but in general, they result in either some type of developmental abnormality or death. And death is actually far more typical than a developmental abnormality, because a lot of polyploidies will just prevent the embryonic growth from occurring at all, because it's just it changes the way that the DNA transcription works in such a profound way that it's just not compatible with life. That question, your 4N chromosome question, Trent, is not answerable because it's not compatible with life. So I can't tell you what a non-existent thing could look like. It's not possible for that to be alive. So as far as I'm aware, so I can't answer that question. Okay, so again, this is just a quick summary of the process here. In order to create life, we fuse two specialized sex cells together. Okay, those are called gametes. In order to produce those sex cells, there are two different processes at work. They look identical more or less, but there are subtle differences between the two. Okay, one exists in males, and that process is called spermatogenesis. Okay, spermatogenesis is the making of sperm. Sorry, I covered it up there with my picture. Spermatogenesis. You start off that process with a spermatocyte. Okay, a spermatocyte is a somatic cell, an adult cell in your body that you find in the seminiferous tubules of the testicles. So they are 2N. And then a spermatocyte goes on and produces 4 sperm cells. Each one of those sperm cells is haploid, or 1N. Okay, so a spermatocyte produces sperm, or spermatozoa. Spermatozoa and sperm is the same thing. Sperm is actually a short form for spermatozoa. But, I mean, pretty much everybody uses the term sperm, so I'm going to use that. So spermatocytes become sperm, and they're produced in the seminiferous tubules of the testicles. The female process is called oogenesis. Oogenesis. I've heard profs pronounce it oogenesis. I think it's just oogenesis, but I've heard people say oogenesis, so maybe it is pronounced that way. I'm not sure. But anyway, it's oo. And that process starts off with a cell, a somatic cell, an adult cell, called an oocyte, or an oocyte sometimes. And that's a 2N cell, a diploid cell. And once it goes through this process of egg production, it's going to produce one single egg. And the egg is called an ootid. So an oocyte produces an ootid. And that ootid is a haploid cell. It only has 1N chromosomes, 23 for humans. But in other species, it would just be half their full complement. Okay, so 2 separate processes here. They look very similar. And in general, this process is called meiosis. Meiosis is spermatogenesis and oogenesis. But oogenesis and spermatogenesis are like the male and female versions of meiosis. So we're going to talk about meiosis in general. And I'm going to point out while we're going through the process what the differences are between when it happens in a male, spermatogenesis, and when it happens in a female, oogenesis. Okay, Kim, does the person inherit the recessive genes but not express them? Yes. So you get one set of genes from your mom and one from your dad. There are cases where one parent's gene for a particular trait will be dominant over the other parent's gene for that particular trait. That gene is still there. It's in the genome of the child, but it's either expressed and not visible, or it's not expressed. So it's one of those two things. Well, again, we're going to talk about that later when we get into gene expression. That's on a different day. Tomorrow we're going to get into gene expression. Actually, no, that's not true. This afternoon we're going to get into gene expression. Is it possible that those same genes are passed down to the person's own offspring? Yes. So there can be a trait that is not showing in you that you got from your mom or dad, and then it may show up in your children because that gene may be dominant in your children, or it's recessive, and then your partner, whoever you have a kid with, also gives them a recessive gene, in which case it will show the recessive trait, whatever that is, because you've both given the recessive gene. Again, we're going to talk about this, how that works later, but good question. Okay, each of the cells that you make are haploid. Haploid, again, means half of the full complement of DNA. I know I'm repeating myself a lot when I say that, but this tends to be something that that students often confuse. And I'm going to use the term haploid and diploid very often in this lesson. And so it's good to be very clear on exactly what it means. Otherwise, it's going to be extremely confusing. And so, again, to get into the exact numbers here, we have a sperm or spermatozoa with 23 chromosomes. We have an egg or an ootid with 23 chromosomes. And they combine together, assuming there's a fertilization event that occurs, to produce a 46 chromosome zygote. Zygote is the name of the cell that has been fertilized. You probably learned that in health class. And it's diploid because you get 23 and 23. So you get now the organism is going to start with the full complement of DNA. Okay. So that's the basic idea of what is happening here, okay? Now we're going to go into the specifics. We're going to talk about how you start off with a oocyte or a spermatocyte and make an ootid or a sperm. It looks on its face very similar to mitosis, which is why it's really important to understand the mitotic process first. And then it's like got a couple of extra things that are added in. It's basically like mitosis twice with some extras. Okay, so again, the overall idea here is that we're making haploid gametes to create genetic variation. Genetic variation means greater survival for a species. That's the theme sort of this. And so as we go through here, you'll notice that I keep mentioning this is where additional variation is created. Okay? There are parts of this process that are crucial for variation to be created in your offspring, new combinations of traits. And variation is important for survival, and so those particular parts of this process are really important. So I'll try and stress them as much as possible. So meiosis is made up of two mini processes, okay? They're split into what's called meiosis 1 and meiosis 2. Each of those looks a little bit like mitosis, but with a little bit of extra changes, okay? So it's similar to mitosis, but it goes through that process twice, which is why it's called meiosis 1 and meiosis 2. The first part is called reduction division. Meiosis 1 is also called reduction division because it separates your pairs of homologs. And by that I mean, remember that you have two copies of your chromosome 1. You have a chromosome 1 from your mom and a chromosome 1 from your dad. And you have a chromosome 2 from mom and a chromosome 2 from dad. The first meiosis is all about separating them so that you've put the one set of them in one cell and the other set in the other cell, essentially splitting your genetic information in half. Okay? So that's what happens during meiosis 1. It's all about splitting up the homologs or homologous pairs. So maternal goes in one direction and paternal goes in the other direction. And it's, by the way, it's not all the maternal one way and all the paternal the other way, because that would mean like all your genes from your dad go one direction and all the genes from your mom go the other direction. That's not how it works. It's a random mishmash of them, but you are going to split them up. Okay, so here we go. Here we go. I also should mention here that this process, meiosis, is often cited as the most complex thing that we discuss in grade 11 biology. So I want to prepare you here. This is the one thing that people tend to be very confused about in grade 11 biology. So hopefully watch the video as an introduction. We're going to go through this process and then we're going to go through it again, okay? Because it is fairly complex, but it's helpful to think of it like mitosis twice. All right, here we go. We start off with what looks like a regular somatic cell. And remember that this is either a spermatocyte or an oocyte. It is the starting 2N cell that you're going to use to make sperm or egg. Okay? And cells begin their life cycle in interphase. And just like the interphase of mitosis, the most important thing that happens during mitosis, or during interphase, is DNA synthesis, replication of your DNA. Okay? The exact same thing happens in interphase 1 of meiosis. Note, by the way, that it's interphase 1 because you're going to go through this process, interphase, prophase, metaphase, anaphase, telophase, twice, 2 times. There's a meiosis 1 and then a meiosis 2. And so it has the same phases once and then again. So when you're talking about the phases of meiosis, there is no interphase. This is always confusing for students. There's no metaphase of meiosis. There's metaphase 1 and metaphase 2. There's interphase 1 and interphase 2, but there's no interphase. Interphase, just the word interphase alone, is mitosis. So interphase 1 is meiosis, interphase 2 is meiosis, but interphase is mitosis. So try and keep those straight. You're always going to need to include a number after the phase when you're talking about meiosis. It's either the first one or the second one, and they are different. Okay, so interphase 1, we make a copy of our DNA. So we're going from a cell that looks like this to a cell that has more of this inside, okay, extra, because you've doubled it up. All right, it doesn't really look like much under the microscope. At this point, you can say that the DNA goes from being 2N, remember 2N means the full complement of DNA, to 2 times 2N. You don't say 4N, because 4N would mean an extra set that is genetically unique to the original set. And as I just mentioned, there is no such thing as a 4n cell. You can't have a 4n cell. So it's 2 times the original genetic information, which is 2 times 2n. Okay, everybody with me here? So if you were to actually go through and count the chromatids, you had 46. Remember that we're talking about chromatids here, not chromosomes, because we didn't actually increase the number of chromosomes because there's no new information. So we had 46 chromatids, pieces of DNA, and then this doubling happened, and now we have 92. Yes, a doubling. All right. Now, prophase is going to happen, prophase 1. This is the most complicated phase in all of meiosis. So we're going to take it slow, okay? Just like in mitosis, you condense the chromosomes into chromatids. Okay, so it's not chromatin anymore, where it's just like spaghetti laying out in the nucleus. We've condensed it down into these visible sticks of DNA called chromatids. Okay, so that's the first thing that happens, and the nucleus dissolves. That stuff is the same as prophase in mitosis, exactly the same. You got condensation happening, and you got nucleus dissolving. We also have, you can just see in the background, that the centrioles are beginning to migrate and form their spindle fibers. Okay, that's happening in the background. That is the same as mitosis, okay? So no difference. The difference here is that now the homologous chromosomes, and remember you have one from mom and one from dad for each chromosome, okay? So let's say that this is chromosome 1 from mom. I did them in pink and blue. Maybe that's a bit stereotypical for coloring, but I don't know, making it easier to identify. Okay, so you got chromosome 1 from dad and chromosome 1 from mom. And they also have their identical partners. So notice that there's 2 of each because remember they got replicated. So they're identical partners. These chromosomes are going to come and give each other a hug. Okay, so your chromosome 1 from mom and your chromosome 1 from dad get together and hug each other. That hug is called a tetrad. Okay, and the process of hugging is called synapsis. So synapsis means that they bind to each other, they grab each other, and then the structure that they form once they have bound to each other is called a tetrad. So a tetrad is really 4 chromatids stuck together. Okay, and keep in mind, I'm only showing you one of these tetrads, but in a human there would be 23 tetrads because you have 23 pairs, homologous pairs, and each one is forming its own tetrad. So you have 23 tetrads. Okay, so they go through and they hug each other. Remember that your chromosome 1 from dad and your chromosome 1 from mom have homologous genes. That means that the genes do the same thing. So they have eye color gene or whatever, or hair color, whatever gene, okay? They're the same on the ones from mom and dad, but they're not identical. So one might be the hair color that's blonde and one might be the hair color that's brown. One might be the lighter skin color, one might be a darker skin color, whatever, okay? So they are not identical, but they are homologs, and that they have the same genes on them. All right, so we have that tetrad that's formed. Then, okay, so homologous chromosomes intertwine and form a tetrad. Then, the most important part of the entire process happens, and that is called crossing over, okay? Crossing over means that parts of your mom's chromosome, your maternal chromosome, and parts of your dad's chromosome, the paternal chromosome, are going to switch. They're actually going to get cut out of one and put in the other one, switched around. Okay? And the reason you do this is you're going to create brand new combinations of genes. It's new variation of genetic material. a combination that has never existed before. And the process is completely random. What gets switched between your mom's genes and your dad's genes? It is completely random which goes where. So, if we were to look at these chromosomes, I'm going to redraw this here to make it a little bit more clear. But we often use letters to denote genes. Okay, so here we have an A gene and a B gene. Okay, and so your chromosome 1 might have a gene that's a capital A. So this might be like blonde hair. Okay, blonde hair, blonde hair. And then the B's might be eye color, brown eye color, brown eye color. All right, that might be the paternal chromosome. Okay, the maternal chromosome might have different genes for those same traits. So it might have little a and little a. So that would be if big A was blonde, maybe little a is brown. Okay, and if big B gene was now I can't remember, what did I said? Hair color? Or did I make the first one hair color? Oh, I'm confusing myself. Normally you write these down. Let's say eye color. If big B was brown, then little b could be blue. Okay, so here we've got the two chromosomes here, your paternal and maternal. When they hug, they're actually going to cut off pieces, and they're not necessarily on the ends, but I'm going to show them on the ends because it's easier to draw. Okay, so they're going to cut off a piece. Oh, that's not what I meant to do. I need a smaller eraser. They're going to cut off a piece, and they're actually going to switch that piece. And then it gets reattached. Shoot, I wonder if I can rotate it. Oh yeah, I can rotate it. There we go. Oops. Come on. There. Okay. So we used to have two versions of this chromosome. Okay, we had the paternal one and then we had the maternal one. But after they've switched pieces between them or done a crossing over event, now we have four completely different chromosomes or chromatids. Okay, so we have this version, which is going to have the big B, big B, big A. Okay, we have this version, which is going to have big A, little b. We have this version, which is going to have little a and big B. And then we have this version, which is going to have little a, little b. So we've created new combinations of genes, or versions of genes. Anyway, they're called alleles. A version of a gene is called an allele. So like the blue hair version and the brown hair version of eye color, or the brown or of hair color rather, would be called alleles. Alleles, A-L-L, I'll write it down here for allele, is the version of the gene. So the brown version would be the brown allele, and then the blue version would be the blue allele of eye color. Okay, does a person is melanin easier to produce, and is that why it is dominant? Wouldn't it just be easier not to not produce melanin, or is there some pushing advantage to more light resistance to UV? Are you talking about melanin in skin color, in melanocytes? Just to be clear, Kim, melanin is not only produced in melanocytes, it's also produced in your iris to make your eye color darker or lighter. So it's not exclusive to skin. Is it easier to produce? It actually depends on the context of whether or not it's dominant. In eye color, melanin production is dominant. So darker eyes are dominant to lighter eyes, like blue. It's in skin color, there is no one single gene that determines melanin production. It's not an on or off switch for skin. there's actually a whole bunch of different genes that contribute to skin color. So we call that a polygenic trait, a trait that has many genes that contribute to it. And we really don't talk about those too much in this course because they're very complex. I'm not sure I can answer the second question that you're asking. Is there an evolutionary advantage to having darker skin? There is in a darker climate, or in a climate with more sun. If you're living in a very hot climate where there's more sunlight, there is a genetic advantage to having more melanin in your skin. It prevents you from getting sunburned. But if you live in a lighter climate, or lighter, I'm using the wrong wording here. If you live in a colder climate, it's actually the opposite. It's a genetic disadvantage to have more melanin in your skin because it prevents your skin from producing vitamin D. So if you have darker skin in a colder climate, it's actually a genetic disadvantage. So it really depends on the climate where you live, and that's actually how natural selection works. It's always the environment that does the selecting. It decides whether something is advantageous or not. Would they cross over in many different places? Yes, they would. So what I've shown you here is the most basic form of crossing over. Is one single point of interaction. They're called locuses or loci. Loci means is the multiple or one locus. The locus is the place on the chromosome. So this is a single locus crossing over event. But in real life, they cross over in a random number. So it could be 0. It is possible for them to have no crossing over events, but there are many, many points that crossing over can occur at, thousands on a single chromosome. So, and it's a random number. So you could have thousands of crossing over events, or you could have 0, or you could have any combination in between 0 and 1,000. So the process is intentionally completely random. And the reason it's random is so that every time you make a gamete, every time you make a sperm or egg, it is completely different than the other sperm or egg. You never make the same sperm twice, or you probably don't. It would be astronomically unlikely for you to make the same sperm or egg twice. So if you have kids, the chances of making 2 separate children that have the exact same genetic traits between 2 parents is insanely remote. It's like 1 in the trillions, because there's so many potential combinations of genes here. So again, this is the process that creates genetic diversity, or at least the key process that does it. There's actually one other thing that contributes here, but this is the real key process that contributes to genetic diversity. It creates brand new combinations of genes. And yes, to get back to your question there, Kim. Yeah, the melanin thing is highly dependent on how it's being expressed, like which trait that's melanin related. Like I said, in eye color, darker eyes are dominant to lighter, that's more melanin production is dominant to less. But then for skin color, it's many genes contribute. Do I have a chromatid identical to each parent and 2 unique to you? Do you have 8? No. You would not even have one single chromatid that is the same as as your parents' chromatid, with one exception. If you are a male and you receive a Y chromosome, that Y chromosome is your exact father's Y chromosome. There is no difference from your Y chromosome to your father's Y chromosome. And the reason for that is that you only have a single Y. So there is no pair to the Y in order to do a crossing over event and create a different version of the Y. There is, you can't mix the Y up, in other words. It's the same Y. So your Y chromosome is just getting passed on as is, if you're a male, to a son. Females are not going to receive a Y chromosome from their father because they got an X chromosome from their father, in which case they will receive their exact father's X chromosome. But their mom's X chromosome that the mom gives to put to both their male and female offspring is going to be a mix, because females have 2 X, which means that they can do crossing over between the 2 of them and create a unique X. So that's the one exception, is that you have one chromosome that came from your dad unaltered, and that's either the X or the Y. But other than that, everything is a mix. Did that answer your question, Anastasia? All the other chromosomes would not be identical to your parents, though, because they mishmashed their chromosomes in order to make the set that they gave you. So you would never have one stick of DNA, a full chromatid, a full chromosome, that was exactly the same as one of your parents' chromosomes. That does not happen, except for the X or Y from your dad. Okay, so again, this is the complicated thing that happens during prophase 1, this crossing over event. The end product of switching genes with crossing over is a chromatid with entirely new combinations of genes. Getting an entirely new combination of genes in genetics is called genetic recombination. So this is one way to get genetic recombination. This is the natural way to get genetic recombination. In the lab, we can break apart DNA in an organism and reassemble it in a different way. We do that all the time. That process is also called genetic recombination, but that is artificial genetic recombination. We can do that in the lab, but this is natural genetic recombination. This happens as part of the meiotic process. And then the last thing I want to mention here, sorry, I've just described what's going on above here. So synapsis and crossing over is a physical exchange of genetic material, and it increases genetic variation. I'm going to mention that again and again and again, because this is what makes us so much more survivable as a sexually reproducing species. We create all this crazy amount of variation in our offspring, making us more survivable as a species in the long term. Okay, so I promise that after this, the steps are simpler. This is the most complicated step of the entire process of all of meiosis. This is the one that there's a lot going on. The other ones are a lot more straightforward than this, especially if you know mitosis. Okay, so I'm going to keep going now, but keep in mind that this is the most complicated step of the entire process, and it is prophase 1. Prophase 1 is like the heavy hitter of meiosis. Okay, here we go. I'm going to keep going. Hopefully we're okay with writing this stuff down, and if not, I do post this. This is posted. So, okay, just like in metaphase of mitosis, metaphase 1 is all about lining things up in the center. But there's a slight difference here. In mitosis, when you line things up in the center, you put the centromere of the chromosome on the metaphase plate in the middle. So I'm going to draw what I'm talking about here. So if this was mitosis, you would have a chromosome, and in mitosis they'd be identical, right? And at this point, you would line this up right on the metaphase plate, right? Because in the next step, you're going to pull them apart, and one is going to go one way and one is going to go the other. But in metaphase 1, you don't want to separate the sisters. Remember, the exact copies are called sisters. You don't want to separate those. What you want to do is you want to separate the homologs. And keep in mind that those homologs are still in their tetrads. Okay, so these are still hugging. So when you line them up, you want to line them up so that the centromeres of each of the homologs are on opposite sides of the metaphase plate. Okay, so you're going to line them up like this. So you're not going to line them up with the centromere on the plate, like in mitosis. You're going to line them up with, whoopsie-daisy, you're going to line them up with the centromeres on either side of the metaphase plate. Because what we want to do here is now separate the paternal from the maternal. And so what you might be thinking is, well, okay, well that's kind of weird. So what we're going to end up with at the end is we're going to end up with all paternal on this side and all the maternal on this side. So you get like a mom sperm and a dad sperm? No, you don't do that. When you line them up on either side of the plate, the side that the maternal part goes to and the paternal part is random. So sometimes you're going to get the paternal on this side and the maternal on this side, and sometimes they're going to be switched, and this one will be over here and this one will be over here. And where they go, which side of the plate they go to, is completely random. It's a random assortment of chromatids, or homologs, I should say, to either end. Okay, and that is important because it means that this is another place where we're going to increase variation. We're going to make the paternal go one way and the maternal go the other, or have it be the other way around, completely random for each pair. So that randomization is called independent assortment. It means the sorting of one chromosome is completely independent from the sorting of another chromosome. So if the paternal chromosome goes left in one pair, the paternal might go right or left in the second pair or the third pair. It's completely independent. That's why it's called independent assortment. So it's completely independent where the particular chromatids will, the homologs will go. Will they go left or will they go right? Random. Okay, so the side that the tetrads are organized is random. That's called independent assortment, and it also increases genetic diversity because it means that you're not going to get mostly paternal on one side or mostly paternal on the other side. It's a mix of maternal and paternal. So I just mentioned here at the bottom that they don't line up on the plate like they do in meiosis. They line up on either side of the plate. I'm gonna take a quick stretch here. Sometimes I spend so much time hunched over my iPad here, my back starts to explode. Whew, hopefully you guys are doing okay at home. These days can be long sometimes on the computer. Oh, I feel that. Thank goodness it's Friday, eh? Woohoo! But you guys are excited to have a weekend. Relax a little bit. Okay, any questions about the process so far here? I realize that we've only made it to metaphase 1. There's really not a lot more complicated left here. It's fairly straightforward. This is a good time for me to point out. At the beginning of tomorrow's class, we're going to do a Google Meet. We're going to try and do this, okay? Let's see how it goes. And I'll let you know when this is coming. Don't worry. We'll jump out of the live stream and we'll go over to a Google Meet. This is just for a short period at the beginning of the class, and I'll let you know when it's happening. Like I'll mention to everybody to move over. But we're going to do a practice session where you are going to try and do this on your desk surface or bed or whatever. You don't have to have your camera on. Sorry, Monday. Yes, Monday, Cedar. Sorry, not tomorrow. I don't do Saturday classes. Monday. So what you will need is something to represent 2 different chromosomes. So you really need 4 pieces of string where you have 2 pieces that are one color and 2 pieces that are another color. Or 4 pipe cleaners, 2 one color and 2 another color. Or 4 really anything that you can cut, rubber bands, twist ties. I'm trying to make this so that you will likely have these materials at home. They can really be anything, but the idea is that you have 2 different colors. So you have 2 sticks that are one color and another 2 sticks that are a different color. Stick, insert whatever you want for stick, pipe cleaner, piece of string, whatever. Okay? The PDFs of these lessons? Yes, absolutely. This is actually posted right now, but I will update it at the end of the lesson. So if you're looking, Trenton, for the PDFs for any of the notes that we've done so far, at the top of Brightspace, there is a heading that says course notes PDF or course notes or completed course notes or something along those lines. And then you click on that and there's a Google folder that has all of the PDFs for all the notes, and that includes this one. So yes. where was I going with this? Oh yeah, and what we're going to do is we're going to do the process together, okay? And then you can ask questions as we go. I'm going to have it open as a Google Meet to help facilitate if people want to ask questions while we're going through the process, because there may be questions in that particular case. It's more likely there will be. And you don't have to have your camera on if you don't want to, and we'll keep the mics muted until you're asking a question, just like you would in any Google Meet. You can have your camera on if you want. It's nice to see people's human faces, but I mean, if you don't want to, it's totally up to you. But anyway, we're going to go through the process again tomorrow. We'll do a review of it because I realize that this is somewhat complicated. So anyway, that's metaphase 1. Keep in mind we're still in meiosis 1. Okay, so we've lined it up in the center. Now let's think ahead. After you do in mitosis, after you do metaphase, after you line them up in the center, what is the next thing that happens? What do you do? This would be what I would call banana phase, but otherwise known as anaphase. What happens in anaphase? What's the main thrust of it? What's the purpose of anaphase? Throw it in the chat. I know you know this. I get the feeling that nobody really wants to type this probably, but come on, interact. By the way, I would like to point out that you guys, this class, asks more questions in the chat than any class I've ever taught this year, which I really appreciate, by the way. It makes the class far more interesting if people are participating. Okay, pulled apart. Thank you, Callum. Thank you. The chromatids are going to get pulled apart. So here, instead of sisters being pulled apart, it's the same idea, okay? But here, instead of sisters, we're going to pull homologs apart, okay? So going to the next step here, anaphase 1, here we have pulled the homologs apart. And you'll note that I've indicated that independent assortment has occurred here because we've got paternal going one way and maternal going the other way. And it's different for the second set. That's right. Now it's homologs being pulled apart. So the sisters are staying together. The sisters do not separate from each other in meiosis 1, in the first half of meiosis. Only homologs separate. So we've got homolog separation. And that's it. There's nothing else, thankfully. This phase is not different other than its homologs. But it is important to mention that these chromosomes, for example, this chromosome, used to be 2 identical sisters, right? But the sisters are not identical to each other anymore because of crossing over. The sisters are actually different from one another, and this is what I would call non-identical sisters. Okay, so they are still sisters, they're attached at the centromere, but they're not identical to each other any longer. Okay, can I keep going here? I'm going to keep going. Let me know if I'm going too fast, please. Okay, then, so you had anaphase. Remember, anaphase is just pull them apart. And then telophase, again, exactly what you would expect in a telophase. This is telophase 1, but telophase is when you put them back in a new nucleus. They decondense back into chromatin form, and then you activate cytokinesis. You can see the cleavage furrow forming there as the beginning of cytokinesis. And the chromosomes decondense and they go back into their chromatin form. So this is just like any telophase. This looks no different than any telophase. Except that we have sister chromatids that are still attached. But the same process is happening. And then lastly, the very end of meiosis 1, we have cytokinesis occurring. So cytokinesis is just the splitting of the cells into 2 cells. In my diagram down here, I just did a little scribble in each of these nuclei to show that DNA has decondensed. They show it as chromatids being in there still, like visible X's, but they're not actually visible during cytokinesis because it's the end of telophase and they've decondensed. So I'm not sure who made this diagram. I actually think it's taken from a textbook, but you can't actually see the X's at this point in the process. So scribble over. There's lots of mistakes in textbooks. And I mention here just the amount of genetic information in each here. They no longer have 2N information because we've split up the homologs, the maternal and paternal, into one or the other. So it's not 2N. However, it's a little bit more than 1N. Because 1N would be if they only had maternal or only had paternal, but they actually have a mix of maternal and paternal due to the crossing over events that have occurred. So I call this 2 times 1N. They have 1N information in them and then another 1N information. So 2 times 1N, but it's not 2N because 2N would imply that they have the full genetic complement that they had, that a normal somatic cell, adult cell, would have. And they don't have that. They don't have the full complement because we separated out maternal and paternal. So I'm going to call this 2 times 1N. In the textbook, they just call this 1N. But 1N is a little bit misleading because of the crossing over event, they actually have slightly more than 1N information in each. So I'm going to call it 2 times 1N. The textbook calls this 1N. Whatever. It's not a huge deal, but I mean, we're playing with semantics here. But also, I think in the note, I accidentally forgot the 1 here on cytokinesis. All of the stages have a 1 on them because they're part of meiosis 1. So this is actually cytokinesis 1. It's the first cytokinesis in meiosis. So if you don't have that on yours, if I haven't fixed it yet, don't forget that. And I think I forgot to add it. So at the end of this process, we have 2 cells. But this is very different from mitosis because the 2 cells that we have are not the same. They actually have different genetic information in them because of the way we've separated stuff out. So that you couldn't just have each of these cells go on and just be their own adult cell at this point. They don't have enough information in them to form a full functioning cell. So we've reduced the amount of genetic information in each cell, which is why meiosis 1 is called reduction division. So it's not a diploid set anymore. It's 2 haploid sets, which sounds like it should be diploid. 2 times haploid should be diploid, but they're not different from each other enough to be a diploid set. It's like 2 haploid sets that are mostly the same, but like a little bit different. So meiosis 1 is all about separating homologous pairs. Am I okay to keep going here? I'm probably going too slow for some people, I would assume. But I do understand that this process is fairly complicated, so I'm not going to blow through it super fast. Okay, I'm going to keep going. Okay, so now what we're going to do is we're going to go through meiosis 2. Okay, so we're starting off with 2 cells. But now the objective here is not to separate the homologs and reduce the amount of genetic information. The objective here is to separate the sisters. So if you'll remember from mitosis, the purpose of mitosis is to separate the sisters, which are identical. So because the objective of meiosis 2 and the objective of mitosis, which is to separate sisters, is the same objective, these processes look almost identical to each other. So that should be a little bit of a relief. If you know mitosis, meiosis 2 is very similar to mitosis. There is one very important difference, and that is in interphase. In mitosis, you make a copy, right, of the DNA during S phase. But in interphase 2 of meiosis, you do not make a copy of the DNA during interphase. If you made a copy here, you would basically start all the way at the beginning again. You'd have too much DNA and you'd have to do 2 more divisions, and then so you'd be restarting the entire process over again if you had a second replication of DNA. And we don't want to make more DNA at this point. We're trying to separate it out so that there's one N in each chromosome, or in each chromosome, in each cell at the end. So there's no replication of DNA. I can't highlight that enough. Many times when students do their project where they create their learning device or their learning aid for meiosis, that's what the project is for this unit, by the way. You're going to create a visualization of meiosis for to teach another student about how the process works. They very often do replication of DNA during this stage. So I can't emphasize this enough. Do not make another copy of your DNA. Okay. So you don't make a copy during interphase 2. Then in prophase 2, it's going to look exactly like mitosis. So in mitosis, you have spindle fibers forming. You've got the nucleus breaking down, and you've got DNA condensing into chromatids, okay? And they're in that X form because remember that the sisters are still attached to each other, and so it just looks exactly the same as it looks in mitosis. But keep in mind that there are only 23 X's inside, not 46 X's like there would be in a somatic cell. Because remember, half of the X's, half of the chromosomes, were split off into another cell. So each of these cells here now have only 23 X's in them. That's important to remember. Okay, then metaphase, again, exactly like it looks in mitosis. You have the chromosomes lining up right on the metaphase plate where the centromere is sitting on the plate, and one of the sisters is on one side and the other sister is on the other side. Okay, and I have 2 of them here because it's happening twice, right? There's 2 cells doing this. Then in anaphase 2, just like in anaphase of mitosis, the spindle fibers are going to pull on the kinetochore, on the centromere, and pull the sisters apart from each other to opposite ends. Okay. Now, telophase is going to occur. And in telophase, you wrap up the remaining genetic information inside a new nucleus, and the DNA is going to decondense and go back into its chromatin form. Okay, so you will end up with 4 cells at the beginning. Or at the beginning. Jeez, I'm having really trouble with language today for some reason. I gotta have another cup of coffee. You end up with 4 cells at the end. If this is a male, okay, so if this was, remember I talked about that there's small differences here between the male and female version of this. If you're a male, each one of those cells will become a sperm cell, okay? So one spermatocyte, that's the cell that you start with at the very beginning, it's called a spermatocyte, is going to make 4 sperm cells, spermatozoa, at the end. Okay, so one cell makes 4. For females, you only make 1 egg at the end. And I'm sure you're thinking, well, what the heck? You made 4 cells. How do you only get 1 egg out of that? I'll explain. So let's say that you're starting off with an oocyte, okay? This is in green here, this is my oocyte. When you get the first cytokinesis happening, that's cytokinesis 1, the first cytokinesis, what happens is that most of the cell contents, the Golgi, the ER, the mitochondria, etc., all goes one way, okay? So at the end of cytokinesis, you end up with a little teeny tiny cell and a big cell that contains virtually all of the cell contents. Okay, so that little teeny tiny cell only has the genetic information in it and a couple other little cell machinery things, but not very much stuff ends up in one of the cells. Okay, and then these are going to split again in cytokinesis 2. And this little tiny cell splits into 2 really, really tiny cells at the end. And this big cell splits into a tiny cell and a big cell. Okay, so they're uneven splits. And so at the end, this becomes the ootid, the egg. And these other 3 over here become what are called polar bodies. So the polar bodies actually just get absorbed. They just get absorbed into surrounding tissue. They do apoptosis, which means they commit cell suicide, and they pack up all their materials, and they basically just give all their materials to the surrounding cells. So they disappear. And the ootid is the egg. So there's a very good reason why it happens this way in females, and that is that the egg does all of the work. So once an egg is fertilized, that's when the sperm, the genetic information from the sperm, goes into the egg, and they unite that genetic information together. So you get 2 N chromosomes inside, male and female information combined. Remember that the sperm is extremely small. Okay, so if this is, I'm going to show this exact same thing, but for the sperm. Let's say the sperm cell starts off, it's actually quite a bit smaller. The spermatocyte is much smaller. And they do their split. And then they do their split again in cytokinesis 2. And then we end up with, those are meant to be flagella, by the way. Okay, we end up with 4 sperm cell spermatozoa. The only thing that is contained in a spermatozoa is just basically, I mean, there's a few mitochondria in there, not many. You need them to power the flagella, so you need a few mitochondria. But it's basically a few mitochondria and a nucleus. That is it. There is virtually no Golgi, no ER, no ribosomes. It is just a DNA missile. That's really all that it is. There's a propulsion mechanism and DNA. That is the only thing that is contained within the sperm cell. So when the sperm cell then comes over and unites, can I highlight this yet? When the sperm cell comes over and unites with the egg, the male version is not contributing any of the machinery that's necessary to build a baby, which is what you got to do once you get those 2 together, right? And so the female actually contributes everything. Thousands of mitochondria, thousands of Golgi, ribosomes, millions of ribosomes. Anyway, all of the machinery is on the female side, because this is the difference in reproductive strategy between males and females. Females focus on quality in their reproduction, They're looking to produce one quality egg with all of the machinery. It's very energy intensive. Remember that this cell is visible to the naked eye. It is an absolutely gigantic cell. So females are contributing all of the machinery for the baby building, whereas the male strategy is quantity. They are producing lots of DNA missiles, spermatozoa, but the quality of them is questionable. And they don't contain a lot of machinery for the actual end production of an offspring. They only contain the DNA. And this is how we determine as biologists which is the male and is the female, because the female reproductive strategy is quality and the male reproductive strategy is quantity. And this is true across all sexually reproducing organisms. That's why for fungi, which do the plus and minus spores, if you remember back to diversity, we talked about they have the plus and minus spores. We don't call those male and female because they are identical to each other except biochemically. And so they don't have these male and female ness strategies to them. They are just all the same. So that's what the plus and minus is. That's just like this and this, but they're mostly the same. But typically in sexual reproduction, you have a male strategy that's occurring in a female strategy. And this affects behavior. This affects all kinds of things. This is why also in nature, typically the females in mating rituals, and this is specific mostly to animals, or exclusively to animals, the females are doing the choosing. Females usually do the choosing of their partner because they basically have a one-shot deal. They make the egg and it's very energy intensive. So they're choosy about who they give their egg to, about who they allow to fertilize the egg. Whereas males in nature, in animals, typically have a quantity strategy for mating. There are lots of species that have long-term mating relationships, so that's not exactly what I mean, but it's typical in nature for males to favor quantity over quality in partners because they're able to fertilize many partners. It's physically possible for them to do that. So that's what tends to happen in nature. Okay, so I just mentioned this here that the female becomes an ooted, one egg and 3 polar bodies because the division of the cytoplasm is not equal. Remember that when you do your project, you're going to do a project where you're going to explain this process. You are going to pick a male or female version. Okay, so you're going to tell me about the male mosquito, whatever, yellow fever mosquito, or the male, what's the one that people always pick? There's a deer that only has a few chromosomes, the muntjac deer. You're going to pick the female or the male, and you're going to tell me specifically about oogenesis or spermatogenesis, not about the process in general, but you're going to pick male or female. Okay, so you do have to talk about what the end product is. Are you talking about sperm, or are you talking about 3 polar bodies and an ootid? All right, so in summary, the process of meiosis 2 is separating sisters, just like mitosis, except there's only 23 in meiosis 2, where there would be 46 in mitosis. And why is this important? This is getting back to the overall important things that are happening during this process. 1, crossing over creates genetic diversity. That's when you get exchange between homologs. Independent assortment, where you decide whether the maternal or paternal are going to go left or the maternal and paternal are going to go right. Okay, that contributes to genetic diversity. And the last thing that also increases genetic diversity is random pairing of gametes, which basically means that which gamete is going to actually make it to fertilize the egg, or which egg is going to be fertilized by the sperm, because some species produce many eggs depending on the species. Like, for example, there are species of octopus that make millions of eggs, so or at least thousands of eggs. So it's not always just one for human females. It's usually one or two. There is a random pairing that's going to occur. So which egg is going to go with which sperm? It's random. Which sperm is going to make it to the end? Which one is going to win the race? it doesn't have anything to do with how good the genetics are inside of the sperm. It's just luck, basically. As long as it has a functioning flagella, whenever one makes it there, it's just luck. So that's random, which is going to pair. So these three things are what contributes to genetic diversity. Great quiz question. I put this on the quiz. But these are the three important places where we produce genetic diversity, and genetic diversity is important because of survival. Okay, we've just gone through the entire process. What time is it? 9:49. Oh boy, I feel like I've been talking for hours. This is a real toughie. There's a lot going on here. What I'm going to do here is do a very quick run through of the entire summary again, but I'm going to draw the chromosomes so that you can see them. Okay, so I'm going to go through that, and then we're going to take a little bit of time together and talk about what happens when this whole process goes wrong, which it does sometimes. It's not perfect. Okay, here we go. In this organism that I'm going to show you, there is a diploid number of 2. That means that the full number of chromosomes that you have in this organism that I'm about to show you is 2. I represent them with 2 different colors. Okay, I don't know if there are actually any species that have 2, but I'm going to use red and blue to denote the colors. Okay, so here we are. Here are our 2 chromosomes. This would be a maternal and a paternal of a particular chromosome. It has to be chromosome 1 because this one only has 2 total. So this has a diploid number of 2. So there's 2 chromosome 1s. Everybody with me so far? Okay, so the first thing that happens in interphase 1 is you make a copy of both of these, right? That's the interphase thing, during S phase, during interphase. So here I'm going to bring my copies in, and I'm actually going to change slightly how I've drawn these. Oh, that's not. Oh, let's make it more obvious. There we go. Okay, so now there's copies of each. That happened during interphase 1. But they are homologs to each other. They have the same genes on them. Then in prophase 1, they're going to condense into chromatids. So now they'd be visible under the microscope. We'd actually be able to see the X's. And they're going to form tetrads. Okay? In their tetrads, they are going to exchange genetic information between the maternal and paternal of each chromosome. Okay? That's called crossing over. We will go through this again tomorrow, by the way, as well. And I'm only going to show one crossing over event, but remember that there are multiple crossing over events that can happen. Okay, so these are actually going to get exchanged. This is during prophase 1. Then, in metaphase, these are going to line up on the metaphase plate with either the maternal on the left or the paternal on the left. Okay, keeping in mind that that's random and it may be different for all the other chromosomes. Okay? Now in anaphase 1, we're going to get the homologs to separate from each other. Okay, now this is going to become irrelevant here. And in telophase 1, these are going to decondense and go back into a separate nucleus. I'm not going to draw them here, but they're going to go into their own nucleus. Okay, cytokinesis 1 happens, and now we actually get a splitting of these cells into 2 cells. Okay, so now we're talking about 2 different cells here. Now meiosis 2 is going to occur. During interphase 2, nothing happens. You do make some cell machinery for the process to help with the division and stuff, but there's nothing visible that's happening. You can't see new DNA being created or anything like that. So during interphase 2, for all intents and purposes, nothing happens. Okay? In prophase 2, the DNA is going to condense, the nucleus is going to dissolve, okay? And that's it. Then, in metaphase 2, you're going to get a metaphase plate, or you're going to get the chromosomes moved to the metaphase plate, okay? And they're going to separate. Sorry, not in metaphase. In metaphase, they're going to line up on the metaphase plate. Then in anaphase 2, they're going to separate. Okay? And then in telophase 2, they're going to go into their own nucleus. I didn't draw it last time, but I'll draw it this time for posterity here. And then cytokinesis is going to happen, and these are going to split into their own cells. Okay? The important thing to note that when you look at the actual genetic information in these, that they are all different. So this chromosome over here is different from this chromosome, is different from this one, is different from this one. We get 4 genetically different cells at the end. And they all have half of the amount of genetic information as you did at the beginning. We started off with a 2n number, which was 2, and now each of the cells has the 1n number, which is 1, half of the 2n number. Okay? When you do your visualization, when you show this to me, this process, obviously you're going to include a little bit more detail, a lot more detail. You're going to include all the detail that we had in the note. But note that the number of chromosomes that we have at the end here is half of the number that you had in the original cell. This is one of the number one things that people get wrong on their assignment, is they start off with a cell that has 6. Okay, that's one of the species you can choose that has 6 chromosomes. And then they end with each of their sperm cells having 6 in them. That means that something has happened wrong, right? Because you need to have 3 at the end. You need to have the haploid number, half of the total. So just make sure that you are actually doing a reduction division so that you have half as many at the end. Okay, so that's the process in brief, and that's with the simplest possible organism. We're going to go through that example again tomorrow at the beginning of class, and you're going to bring a material. You can even use strips of paper, guys. You can use basically anything you want. Strips of paper and like a glue stick or I don't know, even a piece of tape to tape them back together again after we do the crossing over events. Some scissors, a couple strips of colored paper, and some tape. That's all you need. Okay, so anyway, we're going to do that, and I keep saying tomorrow. I forget about the weekend all the time. I mean Monday. All right, so that's the process. Before we move on, I'm going to talk about some errors that can happen during this process now. I know I really filled up the time here during block A. I'm going to give you some time to work on the questions that go along with this during after the break. So we'll talk about them at 11:05 when we come back, because I do want to give you a chance to work on the questions. But I have a feeling that this next thing that we're going to talk about here is probably going to take the next 20 minutes. So unfortunately, I'm talking through the whole thing. The second lesson that we have today, which is going to be on heredity and how we pass genes onto our children, or how your parents pass them on to you, is fairly brief. It's basically just like a little activity that we're going to do together, and I'll do that towards the end of the day. We'll keep that part brief. And then on Monday, we're really going to start digging into the heredity and talking about how do you decide which traits your children get, because there is sort of like a little bit of simple math that you can do to figure out what your children's traits are going to look like based on your parents' traits or on your traits. So let's talk. So any questions? First of all, before we move on, let me know if you there's one part of this meiotic process and you're just like, geez, I am really not understanding blank. Keep in mind that if it's a really complex question, maybe consider dropping in during the Google Meet. Okay, if anything pops into the chat there, I will circle back. Okay, so this, I'm actually going to skip over this. We talked about the impact of crossing over already. This is essentially just reiterating what crossing over is. And I already did that, so I'm going to skip over this. We talked about this idea that letters can be used to denote which version of the gene you have, which allele, and that you get a combination of maternal and paternal alleles in the sperm that you get at the end, or the egg. And then the only difference here is that here they're calling, if you have what is originally in the parent as a combination, they're calling that the parental genotype. And if you get the switch, the one from the crossing over event, they call that the recombinant genotype. But remember that this is a gross oversimplification, because you can have 200 or more sites of crossing over. So there's really no such thing as the parental genotype. I mean, if you looked at the entire genome, you're going to have everything is recombinant. So there's always going to be some form of recombinant genes. And it's not just when you do your tetrad, it's not just like that there's two in the middle that are doing the exchange. These ones on the outside can do an exchange too. So like this one and this one can also be exchanging genetic information. Everything's wrapped around everything else. This is just a simplification for the purpose of making it, you know, easy to understand. But everything is mixing with everything. So I don't really like the way that they've suggested that there's like a parental and a recombinant, because there's no such thing as that really. So anyway, what I want to talk about here. Oh yeah, and then here below, this just talks about independent assortment. Again, we already discussed this idea that if you had no independent assortment, you would end up with this, which is all the maternal would go one way and you'd get maternal sperm, and then all the paternal would go the other way and you get paternal sperm. But what actually happens is that you get a mix of maternal and paternal traits in the sperm because of the randomness of how these get organized. I mean, we already talked about this, so I'm not gonna dig into that too much. What I really want to get to here. Oh man, how many? Is this reiterated how many times? Anyway, this I will mention. These are the three, again, sources of genetic variation in this process. So first of all, you have crossing over, and that increases genetic variation because you create totally new combinations of genes on the maternal and paternal chromatids. Okay, or chromosomes. Independent assortment happens. Again, you're either going to get male or female going one way or the other way. You have, and the reason I'm mentioning this again is because I put the math on here, which is a little bit helpful. You have 23 pairs, right? And every one of them is a random decision, whether it goes left or right. So that means that the amount of different variations that you can get by just organizing them to the left or right is 8 million different possible assortments. It's 2 to the 23rd power. That's the number of different combinations of left and right that you can get. Oh, excuse me. So if you're curious about the actual amount of variability that is created there, it's 8 million possible combinations, and you don't do it the same twice, or you have a 1 in 8 million chance of doing the same thing twice. So it's very unlikely that you're going to do the same sorting. And then lastly, when we're talking about gamete pairing, remember that we're talking about which sperm makes it to which egg. If you have 8 million possible combinations, and we're just talking about the independent assortment here, not the crossing over event, which adds another billion combinations or something to the mix. But if we just talk about independent assortment and gamete pairing, we're talking about 8 million possibilities for the sperm, 8 million possibilities for the egg. So when you combine them together, that's 64 trillion possible combinations. And that's only looking at independent assortment and gamete pairing. That's not even taking into account the crossing over. So the crossing over is adding another billion possible combinations to that. So it's like a billion times 64 trillion. So what are the chances of getting two identical children from the same parents? Well, if it ever happened, I would buy a lottery ticket. It's we're talking about, I mean, in the hundreds or millions of trillions, one in. So in other words, it's not going to happen. And this is just actually a quick summary of the differences between egg and sperm. We talked about the different reproductive strategies again already, so this is just a summary. And this should already be on your sheet here, but females focus on quality. In humans, they only produce one per menstrual cycle. This is human information here, by the way. This is not exactly the same in every species, but the trends are similar. Even in, let's say, an octopus that's making thousands of eggs, sure, the octopus female is making thousands of eggs, but the octopus male is making billions or trillions of sperm. So it's still on an order of magnitude or many orders of magnitude difference in terms of their strategy. So females make approximately one per menstrual cycle. By the way, that should definitely be an approximately, not an exactly one, because it's very easy for more than one egg to be released in a single menstrual cycle. That happens all the time. So approximately one. They're extremely large, 0.12 millimeter, so you would be able to see it. Again, amazing. I think that's so cool. And they're haploid. But meiosis II is incomplete. In other words, females, your eggs that you have, your eggs that you have are not done meiosis yet. They're actually in prophase of meiosis II. So when you go through a cycle, a menstrual cycle, one of your eggs that is in meiosis II is going to finish the process of meiosis II and become mature, and then that's what is released during a menstrual cycle. So they're not actually done all of meiosis. You have all of your eggs. All of the oocytes are already produced. They're actually already produced when you're born, which is a little bit weird. You have all of your oocytes that you will ever have for your entire life are there when you're born. But they don't reach maturity. They reach maturity one at a time, once per cycle. Whereas for males, they're producing spermatocytes for their entire life cycle. So they, starting in puberty, males begin producing new spermatocytes. So there's new ones being produced throughout their lifetime. If you look at I forgot to mention the size here of sperm. So they're extremely small, 50 micrometers. Those are 10,000 times smaller than the egg. So little tiny guys compared to eggs. And you're producing millions of them. This is per ejaculation. This isn't even like per day. The numbers per day are astronomically high. So obviously, again, a difference in reproductive strategy here. And if you look at the insides, I mentioned this already, the idea that female eggs have a huge number of mitochondria inside, hundreds of thousands of them because they have to be ready to produce a baby, whereas incoming the sperm have essentially just enough to get it to the egg. A couple, maybe 100 max mitochondria, and that's just for running the flagella. And very, very little other cell machinery is there. If you look at the special features, there's an egg cell down below there. It has a sugar coat on the outside. It's a glycoprotein coat that it's really kind of cool. It changes conformation, it changes shape immediately after sperm penetrate through that glycoprotein layer. It's instantaneous. And the reasoning for that is it prevents any additional sperm from entering the egg once a single sperm enters. And obviously that's important because if another sperm were to enter, you would get triploidy, 3 of each chromosome instead of 2, and that would be fatal. So then the egg would become non-viable. So that glycoprotein code is pretty important in ensuring that only one single sperm enters. For males, their sperm have a tail, a flagellum, for propulsion. This is for humans again, by the way. Not every male of every species has these features. For example, in plants, pollen does not have a flagellum. You're probably aware of that already. But pollen is essentially sperm for plants, but it has slightly different properties. It has some mitochondria in the middle for energy, very few. And then a little packet of enzymes in the very front of the sperm. They're called acrosomal proteins. And those are what actually interact with the outer glycoprotein layer of the egg cell to allow it to enter. And they activate the change in conformation of the glycoprotein layer. Okay, don't worry about these questions at the bottom. I scribbled them out. You won't need to do them. I think we've probably really beat meiosis to death there. I think we don't need to go too much deeper into that. So what I want to talk about here before the break, oh boy, I got 10 minutes. I'm going to go fast. I want to talk about what happens when this whole process goes wrong. Come on, iPad, don't fail me now. Okay, here we go. So this at the top here, this is just a summary. Blah, blah, blah. Reduction, division, increase in variation. We talked about karyotypes already. Why do I have so much repetition here? That's annoying. This is a summary here of the idea of karyotyping, which we already discussed. This idea that your chromosomes are made up of allosomes, those are the non-sex chromosomes, and autosomes, which are the sex determining, or sorry, I just mixed those up. Allosomes, which are the sex chromosomes, and autosomes, which are the non-sex chromosomes. You have 22 autosomes and one pair of allosomes. And you can organize them by length, centromere location, and banding pattern. This is actually on a different note, so it's not super important that you have that in there twice. Different questions for page 160 on the side of the table. Do you still have to do those? No, you don't. Cross them out. We're going to talk about some other questions you're going to do as a sort of a review for this, and they are not those. So, no, not those. So, what are the problems that can happen here? Well, you can have what's called a non-disjunction event. A non-disjunction is when during anaphase, remember what's supposed to happen is that you're supposed to get, oops, am I going to be able to do this? Let's see. You're supposed to get this happening during anaphase, right? During anaphase 1. That is that one of the homologs goes one way and the other one goes the other way, right? They get pulled to opposite sides. But during a non-disjunction event, what happens is this. Both of the chromosomes go one way. So that's obviously problematic because you're going to end up with more chromosomes in either one of the sperms or one of the eggs. That's if it happens during anaphase 1, because this is an anaphase 1 non-disjunction. I corrected it on your note, by the way. Non-disjunction is actually a one word. There's no hyphen in it. It's one single word, non-disjunction. And then if it happens during anaphase 2, remember that's when the sisters separate, you get this happening where the entire pair goes one direction as opposed to it's splitting like this, which is what's supposed to happen. Okay, hopefully this is making sense to you. So if that happens, so here's an example of it happening during anaphase 1, where both of the homologous pairs have gone one direction. What we're going to end up in the end is to have 2 sperm or 2 at the end of the process for eggs. Remember, only one of these will become an egg. It's easier to talk about it as sperm because not all 4 actually become an egg. But anyway, you're going to get 2 gametes at the end that have 2 of a particular chromosome instead of just 1 of a particular chromosome. And then you're going to get 2 other ones that have none. So they're completely missing chromosome 1 or chromosome 6 or whatever. Okay, so this is what it looks like if you have a non-disjunction in anaphase 1. You could also have a non-disjunction in anaphase 2. So you have normal disjunction. Disjunction is when the splitting, it's the separation and splitting of chromosomes. So when you have a normal disjunction for number 1, but in anaphase 2, that's the second disjunction, one of them becomes non-disjunctive. It does not separate at one of the chromosomes. So now we're going to end up with one gamete that has none, one gamete that has 2, and then 2 gametes that are normal. Okay, so it's obviously less severe if it happens in the second anaphase because it only affects 2 cells out of 4 instead of all 4 cells, which is what happens if it happens in the first anaphase. Okay, so that's called non-disjunction. It is much more likely for non-disjunction to happen in female cells than it is to happen in male cells. And we're not 100% sure why, but part of the reason may be because you carry your egg cells with you your entire life. So the egg cells that you have when you're born are the same egg cells that you use when you are, I don't know, 30 or 21 or whenever you choose to have a child. Okay? So those egg cells have been exposed to the environment, the internal environment of your body for your entire life. Whereas if we're talking about sperm, spermatocytes are produced continuously throughout your life. And so they aren't ever sitting. They're not exposed to the natural environment, the internal environment of your body, oxidative stress, and whatever else might be happening inside your body, chemical stress, depending on what you're eating or taking. And so because they're not exposed to those environmental factors, they're less likely to be damaged in a way that would cause non-disjunction. So it's also true that the older you are, the more likely you are to have a non-disjunctive event. And I should say that males can have non-disjunction. It is possible for non-disjunction to happen in males. It's just far less likely. For females, the older you are, the more likely you are to have a non-disjunction event. So when you're in your 20s, your chances of having a non-disjunction event are pretty remote. 1 in 10,000, I think it is. But once you reach the age of 30, between 30 and 35, your chances of having a non-disjunction event go up fairly significantly. Now it's like 1 in 1,000. And if you're between 35 and 40, your chances of having a non-disjunction event go up significantly more. I believe it's 1 in 500 or something like that. So, I mean, it's still not extremely common, but it's common enough that if you are having a child and you're in your mid to late 30s or 40s, it is possible to have a child in your 40s, it's usually advisable for you to be tested just to make sure that you don't have these genetic anomalies that are happening during the growth of your child, because they're far, far more likely to happen. Okay, so just to show you what this looks like, if you were to take another gamete, so let's say that this is 4 sperm, okay? 2 of the sperm have an extra chromosome and 2 of them are missing. What you're going to end up with at the end, once you add the egg cell in, so now we have a complete set, is that we're going to have 2 that are trisomic. So the trisomic means that you have 3 of a particular chromosome. What you're looking for is disomy of all of your chromosomes, that is 2 of each chromosome. That would be a normal diploid organism. If you have trisomy, trisomy means that you have an additional, a third chromosome for one of the particular chromosomes. Some trisomies are survivable. Down syndrome is a perfect example of a trisomy that is survivable. So you can be alive and have a trisomy. If you have multiple trisomies, it's almost always fatal. So if it happens on more than one chromosome. If you have a monosomy, that is, you are missing a chromosome for one of the chromosomes, this is, I believe, always fatal. I don't know of any genetic illness that is a monosomy that is survivable. Oh, except for monosomy X. Monosomy X is survivable if you only have one X chromosome. But I don't think there's any other monosomies that are compatible with life. I'd have to double check that, but monosomies are generally not survivable. Okay, so we've got 2 monosomies and 2 trisomies here. Pretty much every outcome is not what you're looking for. If you have a non-disjunctive event in anaphase 2, in the second split, you're going to end up with 2 disomies, which are normal. That's what you want, and a trisomy and a monosomy. So here's a 2 out of 4 chance here that you're potentially going to have some genetic abnormalities occurring. Okay, so what are these then? Well, in an organism, when they have the wrong number of a particular chromosome, we call that aneuploidy. Aneuploidy just means the wrong ploidy. So we remember, we're looking for diploid. And so the wrong ploid is aneuploid. Okay, so you either have an extra or a missing one. Both of those are considered to be aneuploids. Again, great word to potentially add to your list. So when you don't have that, when you have aneuploidy, it's gonna cause some problems. I'm really quickly. I'm gonna run out of time. I was hoping to be done this by now. I have another. I think I have another about 10 minutes talking here, and I'm not gonna take it out of your lunch. So when we come back, we're gonna talk about some specific genetic disorders that are caused by aneuploidy. And we're going to talk about some other ways that the process can go wrong. Do I want you to do page 9 and 11? Are you talking about 9 and 11 from the notes? If we haven't covered it in class, you do not need to do it. I jumped over some stuff in the notes that is 100% repetition from something earlier in the notes, and in which case I probably should remove that page. But you do not need to do anything in the notes that we do not cover in the notes. It's not like just like, oh, read it yourself, unless I specifically ask you to do something from the notes. But otherwise, no, 100% skip it over. So my apologies. I probably should have removed that one page. Eventually, just so you guys know, I am going to get you to do some questions from the text that are a review of meiosis. It's page 164, 5, 6, 7, and 11. And there are some questions about errors in meiosis, which are page 166, 1, 2, 3a, 6, and 9. Plus, there is a practice quiz for you to do. So I'm going to get you to do those actually in block 2, and I'm going to give you time in block 2 to work on this. The Mendelian inheritance, which is the last component that's at the bottom there, we're going to do right at the very end of the day. It's sort of like a, it's just like a little introductory activity to Mendelian inheritance, and it only takes about 10 minutes. And so I'm going to do that right at the end of the day, and I'm going to give you time to work on these questions during today. So again, it's 10:20. I don't want to take away from your lunch. If you do want to get a start on any of those questions, you're welcome to do them, but keep in mind that I would just enjoy your lunch. You will get time to work on them in learning block 2. Okay, guys, I'm going to let you eat now. Enjoy your break and stretch your legs. There we go. When you get a both of the chromatids going in a particular direction during anaphase. So it can either happen during first anaphase, in which case both homologs are going to go one way, or it can happen in the second anaphase, in which case both sisters are going to go the same way. But either way, you're going to end up with a polyploidy in some cases, and which is usually a trisomy, or you're going to end up with a monosomy in other cases, once the egg becomes fertilized. Okay, all those are called aneuploidies. So these are common ones, and the reason that they're common is because they're survivable. So it's possible that there are other versions of these that are more common. However, it's difficult to establish that because spontaneous abortion, which is when a fertilized egg, a zygote, begins developing and then dies, is extremely common, and not just in humans, in all organisms. There's a very large percentage of fertilized eggs do not make it out of the first trimester, and I don't think people probably don't realize that. Usually when a miscarriage occurs, people, well, I think everyone feels like they're alone. They feel like they're like the only person that's ever gone through having to deal with that, which is, I think, in many ways unfair to people, because it's extremely common. And So when I mentioned that these are these syndromes that are the most common, it's because they're the ones that are survivable, and obviously the fatal ones, it's difficult to judge how common they actually are. So because nobody's testing to see if there's a miscarriage, and nobody's testing to see if what the genetic anomaly is in that case. So there may be others that are more common, but these are the ones that are survivable. So trisomy 13 is also known as Patau syndrome. It's caused by an extra copy of chromosome 13, trisomy 13. It's the least common of all these common ones here, and it's the most severe, which is probably why it's the least common. It's the least survivable. It usually has a median survivability of about 3 days, which is, I mean, these are really difficult things to deal with for humans. If you have a baby and it only survives for 3 days, that's extremely difficult. You can recognize it at birth because there are certain characteristic structural defects and poor neurology that occurs in babies with Patau syndrome. I think that while the median survival is fewer than 3 days, I think there are some babies that survive up to a year, although I'd have to double check that. But in general, this one is not survivable for long term, unfortunately. Trisomy 18 is known as Edwards syndrome. That's when you have 3 of chromosome 18. This one also has a fairly low rate of survival because it causes things like heart abnormalities, malformation of the kidneys, and there are other internal organ disorders that are associated with it, which can often be fatal because they are important structural organs that are required for sustaining life. So trisomy 21 is the most survivable of these. Well, it's by far the most common in the population, mostly because it is the most survivable. It's when you have an extra copy of your 21st chromosome. So I wasn't sure about this last time. I think I said it was 18 or 21. It's 21 specifically for Down syndrome. So I'm mentioning here, you can either have a complete trisomy, so you have a total additional 21, or due to some kind of unusual translocation error that occurred during DNA replication, you can have a partial additional 21. So you don't necessarily have to have a full additional 21. We're going to talk about some of those other errors in a minute, where they come from. But it is possible to have a trisomy 21 where it's not a full trisomy, so the extra that you have is not the full chromosome. And even if you do have a full chromosome trisomy 21, the actual way that this presents in humans is extremely variable. So there could be very profound cognitive impairments that occur in trisomy 21, or they can be relatively minor cognitive impairments. There's physical growth impairment that occurs often, but sometimes it's very relatively minor. There are, in general, a particular set of facial characteristics that are associated with trisomy 21, but again, they can be relatively minor as well. So there's a great deal of variability in terms of how trisomy 21 can present. Historically, like in the 19th century, people with Down syndrome had a very low mean survival in terms of how long they typically lived. However, more recently, especially because we can treat a lot of the potential organ malformations or other health problems that come along with trisomy 21, in general, their mean survival is much closer to the average survival rate in the population. It's not the same life expectancy as the average person, but it's much closer than it used to be. I believe that the average life expectancy for someone with trisomy 21 used to be 18 or 20 or something like that, and it's significantly longer now. Yes. So short answer to your question, Cedar, if you have a partial trisomy 21, those tend to be much less severe, although you can have a complete trisomy 21 that is also less severe. So it's not as straightforward as that. There are some environmental factors that play a role in how severe, and chance plays a role. So we don't actually fully understand the mechanism at play in terms of what causes a more severe or less severe case of trisomy 21. Although you're right that partial trisomies tend to be less severe. So those are the very common autosomal trisomies that are survivable. Remember, autosome are the chromosomes that are not sex determining, so chromosome 1 to 22. There are also a number of sex chromosome or allosomic trisomies. Remember, allosomes are the names of the sex chromosomes. And these are far more survivable. And there's a reason for that. Your sex chromosomes are just not as important. They're really not. They don't have as many functional genes on them as your other chromosomes. I'll give you a perfect example. The Y chromosome is not necessary. It isn't. I mean, it's necessary for maleness in terms from a secondary sexual characteristic perspective, in terms of it's what determines your secondary sexual male characteristics like testosterone production and formation of male sexual genitalia and things like that during embryonic development. So I mean, it's essential for that, but it's not essential to maintain life. So females, for example, you don't have one of these. You don't have a Y chromosome, and you're fine. You don't need it. Um, there's nothing important on that chromosome that you're missing. So, in terms of sustaining life, it's relatively minor, although it is important for obviously creating the secondary sexual characteristics for men. But the X chromosome, on the other hand, is not as disposable. You do need an X, and males and females, you both have an X, and there are some very important there is some important chromosomal information on the X that is required for maintaining life. So you'll notice that none of these on the sex chromosome disorders are missing both X's. In other words, having no X at all in an organism. That's not compatible with life. So you can't get a no X chromosome and still be alive. However, you can get rid of the Y and be fine. So you will end up biologically female, but you won't die. So that's the first one that's mentioned here, which is called Turner syndrome. It's also known as monosomy X, and it's when you have one X chromosome and that's it for your sex chromosomes. So this individual would be biologically female. They have an X, which is used to create the female secondary sexual characteristics. However, they only produce roughly half as much of those things as they generally would as a female. So there are some physical characteristics that are associated with this and some anomalies. It's generally women are shorter in stature. They have a broader chest, a lower hairline. Their ears tend to be set lower on their head. There's more webbing on the side of the neck than is typical. And because of the reduced capacity to produce female sex hormones, estrogen and progesterone, there tends to be gonadal dysfunction, which means that your ovaries do not cause fertilization, or not fertilization, cause production of mature egg cells. They're non-working ovaries. And it also usually prevents you from having a menstrual cycle. It's called amenorrhea, which leads to sterility. So that's typical for people that have monosomy X. Klinefelter syndrome is an interesting case where you have both X's, as you would if you were female, but you also have a Y, so you have a bonus Y. And because you have a Y, you're going to develop all of these secondary sexual characteristics of a male. So you are biologically male in that case. It is the second most common allosomic trisomy. And because, so this is an interesting case because you're producing the full amount of all of the hormonal signaling and everything that you would see in a female. However, you are also producing the full amount of the same hormones that you would see in a male. And so when you combine those together, you get secondary sexual male characteristics, testicle formation, formation of the penis. However, the large quantity of female sex hormones that are produced usually cause reduced fertility or reduction in the size of your testicles, because there's basically two competing factors going on. and sometimes it can cause sterility, but I think in general you can still produce offspring if you have Klinefelters. It's just more difficult. Triple X syndrome is also known as trisomy X, is when you have 3 X chromosomes. So in that case, you are producing the female secondary sexual characteristics. You have the X, 2 X's, just like a normal female, plus bonus. So when you have an extra of this, there are other things on the X chromosome other than genes that are responsible for secondary sexual characteristics. Again, males have an X. So when you triple some of those up, it does have a tendency to cause some other structural formation abnormalities in embryonic development, which results in things like learning disabilities, delayed speech, delayed language skills. Oftentimes, women in this case have normal sexual development, so they look like any other woman, and you are able to conceive, because when you produce an egg, you are going to produce one egg that has the correct number of X's, and one egg that has too many when you do that. So this one is relatively hidden in the population. It's possible that you have triple X syndrome and you just don't know. You wouldn't know unless you had a genetic test. It's not always immediately obvious from the symptoms that are associated with triple X syndrome. So that one's interesting. That one can be hidden in the population. It's actually, I mean, a lot of these Klinefelters can be hidden in the population as well. I mean, you don't always know that you have these genetic illnesses unless you've had a test to actually look at your chromosomes. The last one on here, XYY syndrome. This one's sometimes called Superman syndrome, although that one might be unofficial. I don't know if that's actually written down anywhere. But it's when you have 2 Y chromosomes, which again are responsible for male secondary sexual characteristics, plus you have an X, which again, men typically do have an X. So here, men tend to be taller than average, although there are no unusual physical features associated with it. So most men, I would guess, that have XYY syndrome don't know. Taller than average, they have normal sexual development, they're able to have children. And this one is relatively hidden in the population. Now, my understanding is that there are other symptoms associated with XYY, because I believe there is an increase in things like testosterone production and things like that, which can have a negative effect depending on the amount of extra production, but they're not necessarily characteristic of XYY. In other words, it doesn't apply to necessarily everybody that has XYY. So this one is also easily hidden within the population. I mean, do you have XYY as a male? Maybe. You probably wouldn't know. So anyway, so these are the very common non-disjunctive disorders in humans, trisomies mostly. You'll notice that there's no monosomies except for X. So monosomy X is survivable, but no other monosomies are survivable as far as I know. Because if you only have one of any other chromosome, it's fatal. Okay, so that's going through some common ones here. I want to mention some other ways that you can potentially have errors occur that can be passed on to your children. So these are types of mutations. A mutation is when you have a change to the genetic material that is, and we're going to talk about the different types of changes that can occur, but they alter the DNA so that it is no longer the same as the original. So we're not talking about a recombination here. Recombination is normal. That happens during meiosis. We mentioned that during the crossing over process, where a gene is exchanged for the exact same gene on a homolog. That's recombination, and that's normal. That's supposed to happen during meiosis. A mutation is when you have an alteration to your DNA where it isn't a perfect switching of genes. Okay, and this is going to happen in a whole bunch of different ways, and I'm going to talk about what they're called here. And I believe this information is on the quiz as well. I'll get you to identify some mutations. So first of all, you can have a deletion mutation. So a deletion, pretty straightforward. it's when part gets deleted. Okay, so if you look at a chromosome, your chromosome did look like this, but then this section got deleted, and now it looks like this. So you're missing a part of the chromosome. And what is the end effect of this? Well, it matters very much what part was deleted. If that part was really important, it could be fatal. If that part is very small or it doesn't include some very important genes, it may go completely undetected, but there could be anything in between. Okay, so most of these mutations can result in profound problems. It could be incompatible with life, or they could cause very minor symptoms for an individual. These types of errors are relatively common during the meiotic process when you're producing egg or sperm. However, the ones that are fatal or whatever are the ones that just never make it to maturity, right? So these, you don't tend to see really significant mutations like this in the population because you just don't survive to be alive with that type of really significant one. So it's usually minor ones that are that eventually make it into the population. Now that's not to say that this happens all the time, but it does happen from time to time. These errors occur during the meiotic process, so it's not totally unusual. And you'll hear about a child having a genetic disorder that neither parent has the gene for. So if the mom doesn't have the gene for the genetic disorder and the dad doesn't have the gene for the genetic disorder, well, it must have arose during a random mutation that occurred during meiosis then. That's really the only place it can come from. So that's where these come into play. So you can have a duplication. So a duplication is when a piece of DNA, like this piece right here, gets copied twice. This usually happens when during interphase 1, when the DNA is being copied, you get some weird reconnection, disconnection of the enzyme that is replicating DNA, and it makes a bonus copy of it. So that can happen. You can have an inversion. So an inversion usually happens during crossing over. When the exchange is occurring between two sides, remember you got this one and this one being exchanged with each other, well, one of them gets flipped and it gets attached upside down into the other one. So instead of being A, B, C, D, E, F, it becomes F, E, D, C, B, A. Now, it's important to note that DNA can only be read in one direction. So when you flip a piece of DNA around, it becomes nonsense. So it's no longer able to function when you flip it around because it's basically like reading a book from the last letter to the first letter. It doesn't say anything anymore when you flip it around. So that is a nonsense mutation. It doesn't you can't use that piece, that whatever that is, anymore. So that's an inversion mutation. Okay, these are all known as single chromosome mutations. They can all happen potentially during crossing over, including the duplication. So it is possible for enzymes to duplicate things during crossing over. And they only happen within a single chromosome. So deletion is happening on chromosome 1, or a duplication on chromosome 5, or an inversion on chromosome 8. Okay, those are single chromosome mutations. It's also possible during the crossing over process. Remember we talked about the tetrads coming together, where chromosome 1 from mom and chromosome 1 from dad come together and form a tetrad. Well, sometimes by accident, you accidentally get the wrong chromosomes hugging, and that can happen too. So you might get chromosome 1 hugging chromosome 8. Whoops. So that's bad. And in that case, if they do a crossing over event, they're not going to cross homologs over from each other. You're going to get basically random weird stuff happening, getting exchanged between two totally different chromosomes that are not homologous with each other. And that's when you get things like this. So these are more than one chromosome mutations. Okay. Insertion is when you have a section of DNA from one chromosome getting moved over and inserted onto or into a place on a different chromosome. So you're taking a section from 4 and putting it on 20. You're taking a section from 2 and putting it on 21, whatever. So that's an insertion. It's interesting to note, this is kind of as an aside, that if you look at the differences between species, often the differences between species can be summed up as a product of these types of mutations. For example, if you look at the difference between chimps and humans, We both have a common ancestor, not that long ago. I mean, I guess it is in human time a long time ago, but in geological time, it's not that long ago. And the differences between them and us is that their species had a founding member with an unusual mutation that occurred. And that may have been the origin for when that speciation event occurred. We're going to talk more about speciation in the evolution unit. But these errors can potentially lead to speciation events sometimes. Okay, I mean, very rarely, but they do occasionally lead to speciation events. So, and they also lead to species becoming more different from each other through potential mutations. Okay, so we got an insertion. A translocation is when you do an exchange of genetic information. This is like a crossing over, but it's happening between two completely different sections. So like a piece of 4 and a piece of 20 do a crossing over with each other. I mean, they're obviously not the same gene, so it's just going to be this random weird switch. Sometimes these mutations have no effect. So if you were to do an exchange like this between 4 and 20, and then you still got 4 and still got 20 in the offspring. Let's say that both of those were the ones that were put in a sperm or an egg, okay, just by chance. Well, you'd have all the information, assuming that you didn't accidentally cut off an important gene when you cut it, which is possible. You may have accidentally severed an important gene when you switched them, but you may not have. And if you didn't, there may be no effect here. However, it's far more likely that you're going to then distribute this one and this one into different egg and sperm, in which case it's going to be profound, the problem that's going to occur there. So anyway, those are the typical mutations that can occur during the meiotic process, which introduces additional new features potentially to your offspring that neither parent has. Most mutations are what we would call negative. A negative mutation means that it creates a product in the organism that is not going to help them survive and reproduce. It's going to be selected against by nature. And that's because imagine that you just have a bunch of words. Okay, you know, like those like word magnets that you can like put up on your fridge. And you have the words arranged in a sentence that makes sense. Okay? What these mutations are like if you just moved some of the words around, okay, moved 2 of the words, or you added an additional word in, but you did it randomly. So imagine that you did it randomly. What are the chances that you got new sentences that made sense or were better sentences for survival than your previous sentences? And I mean, you can already see the problem there. Most of the time, if you randomly move stuff around, you're going to get nonsense, or you're going to get stuff that is not as good as what you originally had. So most mutations are going to end up being negative. They're going to be selected against by the environment. Something is going to work less well. However, occasionally, when those mutations occur, you're going to build something better than what you started with. That is a mutation, a positive mutation, a mutation that the environment is going to select for. Maybe you build a better immune system that it's better at resisting a particular virus, or you build a change in the amount of a particular neurochemical, and that change in neurochemicals is going to alter the intelligence of the organism, or something along those lines. I'm way oversimplifying things here, but those would probably be selected for by nature. They would make you more likely to survive and reproduce, and then evolution is going to act on that change. So we're going to come back to these mutations when we talk about evolution, but this is the basis for evolution, is that these random changes could potentially produce something more survivable than the original combination of genes. Okay, so that's all I'm going to mention here. I'm now going to give you quite a bit of time to work, and I'm going to tell you what I need you to do. 1 second. So first of all, for meiosis, there are some questions about meiosis on page 160, 4, 5, 6, 7, and 11. For the errors component, I'm going to get you to read 162 to 166 in your text. It's actually not that much reading. but it talks about several other genetic issues that arise from non-disjunction and from mutation events during meiosis. So I've kind of just skimmed the surface here of what the potential outcomes can be. 162 to 166 talks in more detail about potential outcomes from errors in meiosis. So I'm going to get you to read that and then answer questions on page 166, 1, 2, 3a, 6, and 9. Some of that's from the reading, and some of it is from what we discussed together in the note. These questions, by the way, are what goes in your question document for this unit. Okay, so there you go. Here's the one that goes in your question document, these questions. When you're done all that, there is a practice quiz. There it is, the link to it right there, which are just questions about meiosis and errors in meiosis, just to give it a shot and see what you remember, what you might want to consider going back and taking a second look at if you're not sure about stuff that's in the quiz. And if you have any questions, you can always ask me. Okay, so we're going to work on that till about 12:30. So that's about an hour. And then we're going to come back and we're going to talk a little bit about inheritance. So I'm going to close the stream down here for a while, and we're going to come back at 12:30, and we're going to do a little experiment together. And by experiment, I just mean we're going to assess our genetic traits and look at what our physical manifestations are of our genetics, of our own genes. And look at how common those particular genetic traits are in the human population. Okay, so we're going to come back and do that at 12:30, and I'm going to give you an opportunity to work. Let me know if you need anything in the meantime. All right, so we mentioned previously that you have two vert copies of each gene, right? So you've got a paternal chromosome 1 and a maternal chromosome 1, right? And each of those would have the same genes on it. So let's say that it was for eye color, okay? Maybe it has blue eye color on one and brown eye color on the other. Okay, you have two copies of each gene, one from your mom and one from your dad, okay? That were combined together in the zygote to make your genetic code. Okay, and you can say this for anything. So if you were to write down the combination of genes that you got from your parent, you would write it down like this. So let's say you would have this version of the gene and this version of the gene. Versions of the gene, by the way, are called alleles. I'm sure I have this written somewhere else in this note, but just as a, so these are alleles, versions of the gene. And if you write down both of your versions together, like this, we call this your genotype. Okay? Where are these notes? They are, good question. They are, if you go to Learning Block 2 here, this is today's tab on Brightspace. It says you will need the part 3 of the course notes. Sorry, I probably should have mentioned that. That is these. And we're going to use this, this note package here for the rest of genetics. It has the rest of the genetics information in it. So I'll give people a second to download that and open it in Cami. Sorry. Should have mentioned. Okay. Oh, this is going to work. Yes, it is. No, you don't hand the notes in. Only the homework documents are going to get submitted. So it's the assignments and the homework documents, not the notes. The notes are for your reference, and you can use them to complete the quizzes. They're there as a reference for you. The better notes you make, the better reference you have for when you're writing the quiz. So they're really there to help you. So I'm just giving you a bit of terminology here. So this is a genotype. It is your combination of alleles for a particular trait. This combination will produce in the organism through your expression of the genes. So that's through transcription and translation, right? That was the process that we talked about yesterday, is going to produce a phenotype. The phenotype is what you look like. It is the expression of those genes. It's not just looks. I mean, it could be intelligence or any genetic trait. So that is what you're going to end up looking like. And in this case, we use the capital B for, oh, I should have written what these were. I'll add it in. I think I said that this was brown and that this was blue, I think. So in this case, capital letters denote what is called a dominant gene. A dominant gene is always shown, no matter what. A recessive gene, like the little b here, is only shown if you have both alleles recessive. So if I had two little b's, I would end up having blue eyes. But if I have one big B or two big B's, then I will have brown eyes. So in this case, the phenotype here is going to be brown. Okay, so that's the outcome, and that's known as the phenotype. Great words for your vocabulary list, by the way. These are core. This is core terminology for the unit. So your genotype makes your phenotype, and you have two copies of each gene in your genotype. So I mentioned this, I wrote it down here, but a capital letter shows a dominant allele, and a small letter shows the recessive allele. For phenotypes, if you have two capitals, two dominants, your phenotype will be dominant. If you have, I should probably highlight as I go here so you know what I'm talking about. If you have two dominant alleles, you're going to show dominant. If you have a dominant and a recessive allele, you're going to show dominant. And if you have, two recessive alleles, then you're going to show recessive. That's how you determine the outcome, the phenotype, from your set of genes called the genotype. And that's basically what's just summarized above there in the introduction. It's just that. So what we're going to do right now is we're going to look at some single gene traits in humans. Now, it's really important to note that most of your traits, most of your phenotype, is not determined by one single gene. Most of it is determined by multiple genes. Those are called polygenic traits. So for example, your height is not determined by one gene. There's like a whole bunch of growth hormone genes and genes about the expression of cell reproduction factors and how large your growth plates are, again, which is also genetic, and just other genetic things, plus your environment contributes as well. So those are called polygenic traits that a whole bunch of different genes are going to contribute to a trait. For example, there's no like gene for intelligence. You can't like go in someone's genome and be like, oh, they have the intelligence gene and it's turned on. That's good. It doesn't work like that. There are traits that have only one gene, and then that will actually have an outcome on the organism that is clear, an on or off or a yes or no. And those types of genes or those types of traits that have one single gene are called Mendelian. So polygenic traits, the ones where many genes contribute to a single trait, are very complex in terms of analyzing their expression. How do you know how tall someone is going to be based on their genetics? Well, you need to look at the input of, 55 genes, a lot of genes probably. And so that's a complex process in determining how those two things are connected to each other. Way, way beyond the purview of a grade 11 biology course. We're talking about like advanced genetic analysis that you would do towards the end of university if you specialized in that. So it's definitely outside the purview of this course. We're not going to talk about polygenic traits in this course. So I'm going to be just only discussing, for the purposes of this course, Mendelian traits. Mendelian traits are single gene traits where one gene does one thing. I have the gene for this. I have the gene for this. This is a Mendelian trait. Okay, so just to keep that in mind, most human traits are polygenic, and there are very few that are Mendelian, but we're going to discuss the Mendelian ones, or at least some of the Mendelian ones now. That's true for humans. For plants, plants have actually quite a few Mendelian traits, which makes them much easier to study for genetics. And there are other organisms that have many Mendelian traits. But for humans specifically, and actually mammals, vertebrates in general, vertebrates are very complex genetically, and they have very few Mendelian traits. So anyway, it's just really important to note that we're talking about a specific subset of genetics here, that is where one gene is equal to one trait or one phenotype. All right, so keep in mind that for each of these things that we're going to discuss, you have two alleles, two versions, two copies. Now they could be the same allele, or they could be different, okay? If they're the same, so let's say that you have, like going back to my B example, if you have two alleles that are the same allele, okay, we call that, so if these are the same, we call that homozygous. They're the same. If they're different, we call that, oh, shoot, I have to write different there. If they're different, we call that heterozygous. Okay, so the same alleles, you're homozygous, different, you're called heterozygous. And for homozygous, you can be homozygous dominant, which means you have two of the dominant, or you can be homozygous recessive, which means you have two of the recessive. Okay, so there's just a little bit of terminology here as we go through. I'm going to use these words, and I don't want to like go over everybody's heads. So what we're going to do is we're going to go through a list of genes, or a list of traits in humans, and we're going to determine what your genotype is, or at least as much as we can. We're going to choose what your genotype is. So I got to delete all the data on this here, because this is mine. So I'm going to do my own on here. And normally when I do this in class, last time I did it in class, we looked into the percentage of people in the class that have this trait, but it's really, really hard to do over the chat. The last time I tried to do this over chat, it was a complete failure. So we're gonna leave part of this information out, but I'm gonna discuss how to determine what your genotype and phenotype is for some traits that are Mendelian in humans. Okay, so let's go through our little questionnaire together here. How much time do I got? Oh, didn't leave myself enough time for this. Okay, tongue rolling. We may have to pick this back up on Monday, and that's not a big deal. Tongue rolling. What that means is, can you stick your tongue out and make it into a roll, a U, like this? But contrary to what it shows in this image, you can't use your cheeks. So you can't like push on it with your cheeks. That's cheating. So can you make your tongue into a U? If you can do that, it means you actually have an extra muscle that runs across the width of your tongue, and that is a Mendelian trait. One gene turns that on. Okay, so the ability to roll your tongue, that means you can do the tongue roll, which I cannot. I could try, but I can't do it. If you can do the tongue roll, you have the dominant trait, which is shown with R's. They've chosen R here to indicate the dominant trait in this case, okay? So I personally, this says your phenotype. You need to write in what your phenotype is. I'm not a roller, okay? So but you could be a roller. You write in what you are there. Okay, because I'm not a roller, it means I do not have the dominant trait. So I don't have a capital R. I only have small r. So my genotype is this. That's what genes make that phenotype. Now, if you do have a tongue roller, you're a tongue roller, that means that you are either this, you're either capital R, you have two tongue roller alleles, or you are big R, little r, okay? Those are your possible genotypes. And another way to write this is you can write big R, and then blank, that means that the second one is unknown. So you can actually write it both ways, whichever way is more comfortable for you. But you do have 2 copies. So if you are dominant, you are one of those 2. If you're recessive like me, which is that you are not a tongue roller, you're going to be little r. So I'm going to write my little r here because that's me. I'm writing my own. Okay. And normally I would do percentage of the class. I have a feeling this one is particularly rare. We would normally survey that, but it's too difficult to do in the chat. I did it last time. It takes forever. So, so you write down what you are. Okay, that is a Mendelian trait. I'm gonna do maybe 1 or 2 more here, and then I'm gonna let you guys go and enjoy your weekend. I will say that for the questions here, whoops. You actually have all the information you need to do the practice quiz for Mendelian inheritance. What we just covered in the introduction there is everything you need to do the quiz. So you could, let me just jump over to it real quick here. If you want, you could go and do the exit quiz here for heredity for the first little bit. You don't have to. If you want to leave that until we finish this survey on Monday, you're welcome to do that. But you have everything you need. You could potentially do that right now. This video on heredity, I'm going to ask you to complete during Learning Block 3. It's really the only, other than adding vocabulary to your list, it's really the only thing that you should do during Learning Block 3. So this is going to review that concept of genotype and phenotype and give you a few excellent examples of Mendelian traits. So it goes over it a little bit, and the refresher is good because I realize all this terminology is brand new for you. So I'm actually going to leave it there. Instead of doing another trait together, we're going to finish analyzing our traits, some Mendelian traits in humans, on Monday. I recommend that you do the exit quiz because you do technically have everything you need. But if you want to do it on Monday, it's no big deal. But you should watch this video. It's a good summary of the introduction that we just did on, it's mostly terminology, on hereditary traits. Okay, so I would watch this video. If you have any of the other questions from before still left to do, finish those. You don't have to do the exit quiz, although again, you could do it if you wanted to. You definitely know everything you need to know, and you should add to your vocabulary list. So that's it for me for today. Is the quiz just for practice? Yes. All of the practice quizzes on here that say exit quiz, those are just for practice. So the last one that you would have done, if you did the one for errors in meiosis, the one right there where it says exit quiz, those are just for practice so that you can be aware of the fact that you either understand a concept or you do not understand a concept. If you don't understand why something is wrong in one of the quizzes, that is work for you to do. You need to go back and figure out why you don't know how to answer that question properly. Or you just come talk to me during Learning Block 3. That's what Learning Block 3 is for. And I would be happy to go over that with you. So there are exit quizzes now, I think at the end of every single section. I've gone through and went back and made them. And they're just little checkpoints for you to check on your own understanding. Do I understand this or not? If you're acing every one of the quizzes, it means you probably have a fairly good understanding of what's going on. If you're not, then you just, there's something you have to do about that. I mean, if you're interested in doing well in the course, you have to go back and analyze why you're getting those particular questions wrong. And they will help you out greatly on the quiz if you do that. It'll kind of set you up for success on the quiz. That's the idea. Can you go over why there are more types of trisomy disorders? Kim, can you expand on that question a bit? Why there are more types of trisomy? You mean more types of trisomy than there are monosomy? There are more types of trisomy because if you're overexpressing a particular gene, that is, you're building too many of a specific protein, I mean, often it's fatal. So most of those are fatal. But in monosomy, you're not making enough of a particular protein. And usually, if you're not expressing enough of a particular protein, a biochemical process just does not function. It doesn't work at all. And so if you start severing, the functionality of any biochemical processes, you're going to start getting a bunch of down the line processes that don't work. It's like a domino effect. So if you have the biochemistry isn't sufficient to get this reaction to work, then these 10 things don't work, and then these 100 things don't work. So a monosome usually has this like cascade effect where it causes a whole bunch of different processes to malfunction. Whereas if you have too much, you may get excessive production of certain things, but the basic biochemical processes can potentially still function. But keep in mind that most trisomies are fatal as well. So having overproduction can be survivable, but usually having underproduction isn't, because it just then your biochemistry doesn't work in your cells. You're not making enough of a particular protein or enzyme or whatever, which causes non-functionality. I hope I answered your question there, Kim. So I'm going to let you guys work on whatever you need to work on. Drop by the Google Meet if you need any help with any particular thing. But watch this video during Learning Block 3. It's an excellent review of the introduction there to heredity, and we're going to move extremely fast in heredity. So it's good to have a good foundation for these concepts. There's quite a bit of material. This is usually the longest unit, and we're doing it in about 5 days. So 5-ish, 6 days. So it's going to require some quick movement. Okay, enjoy your weekend. Huh, you made it. It's Friday. Hopefully we'll get a couple of days of nice weather. Guys, take care of yourselves, and I will see you on Monday if you're not going to join me in the Google Meet.